it's my pleasure today to introduce our speaker, who's coming from pretty nearby. Dr. Vaughn Shirey coming from the McGuire Center. He's a new curator. I think he just started in January, so I'm officially not like the baby around here now. No, but yeah, Vaughn comes to us by way of Georgetown, where he did his PhD, I think, and before that Drexel, and does a lot of really cool work on Lepidoptera and pollinators. He's published in places like Nature, Ecology, and Evolution, and also does a lot of modern biology, I would say, in terms of using machine learning and image recognition, or even things like community engagement through iNaturalists and studying how to engage citizen science that way. And so I'm hoping today he'll tell us a bit about that and also what he's doing here now at the museum. So,
Yes, thank you, Andrew. It's a pleasure to be here too. I was told to ask the people on Zoom if they can hear me and see the slideshow first. So if someone just wants to throw a confirmation on the chat before I go. Today I'll be talking mostly about historical ecology and AI, and I normally don't start my talks with a thank you slide, but it's a weird time to be an assistant professor. And so this is my lab. They keep me all sort of sane, and they're pushing my research in a lot of new areas, which has been really great. What do we do briefly? So my lab is really sort of an interdisciplinary ecology lab. We touch on traditional evolution, we touch on ecology and conservation. And then I'm really excited, I'll talk a little bit about this, visual culture and humanities. And through all of that, we really use this data science lens. So we're big fans of math and statistics that I know people think is scary, but it's really not, and it's really sort of an exciting storytelling device. So brief outline of what I'm going to talk today. I'm going to kind of look at my past, so where I was coming from, which was Bayesian historical reconstructions. So looking at asking the question of what the historical butterfly communities look like over multi-decade timescales. And then I'll move into some AI applications and finish out with some cool visual studies stuff. So I'm a global change ecologist broadly, and I think global change ecology is sort of this strange existential discipline to work in. It can become very overwhelming all at once, but in my lab, we use a sort of simple three-pronged framework to think about global change dynamic. And so you can imagine that there's some species that is faced with a changing environment or an environmental stressor. And typically the first response that that individual or species is going to have to that stressor is going to be to move, right? It's typically the easiest response. They'll move by changing their distribution or by shifting phenologies, for example. If that's not happening, we can talk about species maybe adapting. And I'm sort of using adaptation as a broad umbrella term here. So meaning adaptation and acclimation to the stressful environmental conditions. So that includes things like phenotypic plasticity. And then the sad fact is if one of those two things doesn't happen, usually that organism or that species will die. Right? And so in theory, we should be able to pick up signals of these three processes by looking at historical specimen data, for example. And historical specimen data is abundant, right? So we have billions and billions of insects collected that tell us the when, where, and what about where insects have been throughout history. But I always advocate that these data, if you're looking at a historical ecology lens, should not be used solely by themselves or off the shelf, right? We know that for most purposes, they were not collected with the intent of cataloging a community, but rather for something like systematic. And so I like to frame sort of collections in the purely ecological sense as having big data, but little deeper knowledge about process. And so it's up to us to sort of reconstruct that. And so some early work that I did focusing on these sort of big data problems involved having a look at inventory completeness for butterflies across North America. So here we overlapped expert range maps with these 100-kilometer grid cells. And then we looked at all the data from iNaturalist, from things that have been digitized in museum collections. And we looked at between 0 and 100%, how much of the expected species richness was cataloged in those locations. And one of the things that you see immediately is that we're really good at sampling where we live, right? And so these sort of large metropolitan areas tend to have 100% inventory completeness, so we fully documented the expected species richness in that area. But then we have these large swaths of land in North America where we have very little overlap with the ranges, or in some cases, no currently digitized butterfly data available. And when we intersect this map with the velocity of climate change into the year 2100, we see this sort of desperate picture emerge. So although climate change is happening sort of in the lower 48, we see that there's low climate velocity here in the white and full sampling completeness. So climate's not changing as fast in those areas. But when we look at the areas where we haven't sampled historical butterfly biodiversity, where climate is changing most rapidly, we can't really start to construct the story about those regions. And so I became really interested in these sort of high latitude regions because they're experiencing extreme climate change, but not a lot of historical collecting has occurred there. So could we potentially piece together a story about how butterflies have responded to climate change in the boreal and Arctic? And I'll just mention that these data, this sort of adds more complexity. We have a whole bunch of people who are engaging in recording butterflies in different ways. And so a lot of folks are targeted samplers. So they might be, in this case, people who just go out and they want to look at orange butterflies, for example. And then we have folks who go out, and this is the people that I like, are the community samplers. And so they go out and they say, you know what, I'm going to go to a place and I'm going to record everything that I see there, or at least make some effort to. Okay? So that's underlying all of this sort of data paucity. So a big question for me is then, given all this complexity, how can we cut through all of these biases to reconstruct the history of butterfly communities in the area? And so we use this framework called an occupancy detection framework to do it. It's basically a two-level model. And I'll go through that now. So we worked on basically testing whether or not this occupancy detection framework could be used to reconstruct historical butterfly ecology. The first question, which is the question that we're interested in, is the species actually here? Like, what is the probability that in 1970, the species was in Gatesville, Florida? We then layer on top of that, how detectable was it in that time? So the two-stage process, probability of it being there, probability of us detecting it, digitizing it, collecting it, etc. And those two processes are the process by which any data ends up in a natural history collection or on iNaturalist, for example. And so if we can use this sort of process model, maybe we can get at this underlying ecological question. And of course, because this is situated in a mathematical framework, we can then outfit this model with different predictor variables. So we can say, well, I think the probability for the ecology is based on something on maybe temperature, precipitation, and maybe detection. Traditionally, we would use sort of number of observers or party hours in the field. I will say that these models were developed for what we call detection, non-detection data, and we don't have that in museum collection. We don't have the zeros, more or less. And so what we do is we rely on imputing those non-detections. So we basically make an assumption about when collecting was sufficient enough to say things were sufficiently looked for to establish zeros. And this goes back to that sort of dichotomy of people, right? So if I have a targeted collector who's only looking for yellow butterflies, I have weak or limited evidence that they looked for everything else, right? But if I have a community collector who is looking for everything else, then I can maybe impute the zeros for some of these other species. And I'm displaying this sort of on an individual, personal basis, but it can be an emergent property of the system as well. So totally unrelated people who don't know that they were collecting in the same area could fulfill this role. And it's also not binary. We have people who fall very high on this spectrum versus very high. And so to do this, we basically simulated several sort of hypothetical species on a hypothetical landscape, probably about 40,000 simulated trials. We shot a whole bunch of holes in that data to replicate sort of the museum collection process. And we put this up on the Compute Canada infrastructure and asked, when can we break these models? Like, what are the assumptions and the limitations of doing this historical construction? So one of our key findings is that these models, they're robust enough that they perform as well as if you had the traditional data for these models, so long as those community-focused sampling events occur greater than 50% of the time. Okay, so it's super robust. It's as good as if you went out there and had a structured sampling program. So you can apply these really readily, museum specimen data. And then basing non-detection, so the zeros on species seen in the same area at the same time, is a sufficient assumption for allowing these models to work. And so really, it sounds kind of insane when you think about it, but we use just the sighting of one other species in the same area to impute those zeros, and it does not break the model. So what does this look like in practice? This is obviously results from a simulation, but how does it look like it applies to real data? So again, I was really interested in this sort of boreal Arctic region of North America. And we ran this model on about 95 species. This is called a spaghetti plot. Notice all the gray spaghetti in the background. And here on the x-axis, I'm plotting species responses to changes in average minimum temperature. And on the y-axis, the probability that that species is going to be present in that site. So all of the species are my spaghetti in this model, the gray. So you can see that some species respond positively, some negatively, some have these sort of interesting quadratic relationships. And then I've summarized species by whether or not they're warm or cold adapted. So the red line illustrates the average response for those warm adapted butterfly species in the Arctic. So there, as those minimum temperatures increase, we see that increase in their occupancy probability. Nice to have that sort of sanity check. The black is your average butterfly. So again, we see that probability increase with warmer temperatures. As ectotherms, that kind of makes sense. And then the blue are those cold adapted species, right? So as those minimum temperatures are increasing, we see them go from about 100% occupancy probability down to about 25%. Okay. Now, for context, temperatures in the Arctic are projected into 2100 to increase by about, in some places, 10 degrees Celsius on average. So you can sort of look at this scale and see that there's actually going to be a huge shift in some locations. And that will possibly determine future butterfly communities as well. More recently, I sort of dipped my toe into moth world, and we're looking at sort of using these models to understand the combined effects of climate change and urbanization on moths in the northeastern United States. And what we're finding is that there's a variation in scale. So we ran these models at different scales, all the way from 15 kilometers to 75 kilometers. And what I'm plotting here are the effect estimates of the various variables or predictors that we put in the model. So I'll walk you through this really quick. Here's our smallest scale model at 15 kilometers for our 75 coarse model. The green here, so this is the quadratic response to temperature. This is a linear response. So it appears that temperature is the major structuring factor determining distributions across scales. We have precipitation, which remains kind of consistent, although maybe not as important at this glacier scale. And then we have two built environment factors. The light pink is artificial nighttime light, and the dark pink is impervious surface cover. And what we see is that there's sort of this contingent of the same species that are consistently affected by artificial light, no matter what scale. And that the role of that impervious surface cover becomes more and more important as we get to finer grains of analysis. I love maps, so we decided to map this out for a very charismatic species, Polyphemus. And what we're looking at here is percent change in occupancy since 1993. So all of the purple squares are where that probability of occupancy of the species have actually gone down since 1993. And it's gone up where those green squares are. And what you see for this moth in particular is that it tends to be what I would call anthrophilous. So it's sort of increasing around these major metropolitan areas. Now, why is that? Well, we think that there's probably an attraction to light coming there, but that if you know something about the natural history of the species, is that they do not have to pupate underground. And so their host plants are commonly planted as street trees in cities. They're not affected by impervious surface cover at all. And so these cities sort of become these interesting refuge for polyphemus moths. We've also, and this is the biggest study I've ever done, run this on European invertebrates in general. And so this analysis included about 1,000 species of randomly selected invertebrates across the European continent. And here we wanted to reconstruct trends in occupancy probability since 1970. And so there's two distributions here. This distribution that sort of has a lot more variation is a model that's solely informed by time. And so that's why there's a lot of variation to underlying spatial heterogeneity predictors. And then the environmental model includes things like climate, land use, and all of the groups here are organized by losers to winners, more or less. So these groups represent the species that are sort of most in decline across Europe, whereas the groups over here represent the species that are most sort of increasing or at least stable since 1970. And so we've been trying to tease this apart with different traits, body size, things like that. And across the European continent, the single most predictor of whether or not you're going to be a winner or a loser is actually your adaptation or acclimation to precipitation. So Europe, there's a lot of desertification going on, and the more arid tolerant you are, the more likely you are to be on the winning side of local change. Okay, so I'm going to pivot to AI, but I come from a department where people just like blurt out questions in the middle of your seminar. So I'm used to that. So I'll pause here and see if there's like one question. Yeah, Chris.
So on that graph you just showed us recently, not this one, which is cool, I have questions about that.
Yeah.
Later. If you go back, there was a little uptick. With temperature there.
Yeah.
What's that all about?
Yeah, so a couple of interesting reasons. It could be an artifact of sort of averaging across the species, but I think it's actually sort of a non-linear effect. So what's really important for butterflies and insects in general in the Arctic is this area called the subniveum, which is between the snow and the ground in winter. And that's actually several degrees warmer than sort of the ambient air temperature. And so what I think is happening with this is that they're losing that subniveum as the temperatures increase, that snowfall is converting to rainfall, and they don't have that nice blanket in the winter. So they're exposed to cold. But then when those winters become mild enough, there might be another uptick, right? So that's something that I'm really interested in exploring further. And it's relatively, I mean, we don't think of winter as like the most important time for a lot of insects, but up there it's certainly is. Okay. Sweet. So I'm going to switch gears really quick. Sorry for the whiplash. To AI and deep learning, and I think like being at UAT, that's like all you hear about, sort of. So I'll talk a little bit about kind of the things we've been doing in AI, particularly computer vision, and then some of the stuff that I'm really excited about. Right now, I'm going to talk about some work that was actually done in Akito's lab by one of his PhD students who I helped out with her thesis, focusing on this autonomous group of moths. You can see very striking morphology. It's sort of been written for decades and decades and decades that these sort of mimic the eyes of owls. And so we wanted to investigate whether or not there was a pattern matching between this morphology and the actual faces of owls. What we can get from images, as we use iNaturalist images, of course, is the taxon, the location, and the time. But more and more, and I think what's increasingly exciting is that we can get sort of these more niche information about environment or microhabitat or biotic interactions from iNaturalist photos as well. Right, so here's, I've sort of inverted a cropped out cutout of this autonomous hindwing, and if you squint, you might have thought of an owl. But here is an owl face. I really love this comparison because I call it the blue eyeshadow moment. So there's like blue eyeshadow on the out, and there's like blue around the, anyway. I was trying not to like bias myself on this too much, but the model more or less goes that the autonomous has an anti-predatory defense because what's eating those moths most often is really afraid of those owls, right? And so it's sort of this sort of trophic alignment there. There was an ancestral state reconstruction done here. I'm showing the complexity of the rings around those eye spots on the hind wing. And you can see that there's sort of this evolution of complexity along the tree, going from no rings to some in some groups to sort of more complex concentric ring structures. And then we call this the glimmer. And the glimmer is this white part in the sort of center of the eye spot on the wing that is sort of, at least we think, supposed to replicate sort of the refraction of light within the eye of the owl. And so you can see that sort of the outgroup here does not have the glimmer, but then most of the species in Alzheimer's does have at least some of that glimmer. Can be nice. So again, here's our nice case study with some of the best images that we got from my naturalists. We wanted to basically feed both of these images into neural network simulations. So we fed them to neural networks. We asked the neural network to classify those images as either an owl or a moth, basically on one look of the image. And then also tell us a little bit about the patterning on the wing and or on the moth or the owl face, about where the neural network is being used. And so again, we wanted to say when and where do these neural networks confuse by the moths and the owls? And then do extracted features or patterns correlate with the overlap or sympatry between these two species? Now, I'll just say very carefully that we're not saying that this is a brain. We're just looking at it to extract sort of these high dimensional features. So here I'm showing that same pair of moth wings. And this is what we're calling an entropy map. And so these sort of green areas are where that particular model is getting confused and saying, these are areas that are tripping me up and making me want to classify this as owl instead of a moth. So we get these sort of like highlighted areas around the concentric circles and maybe also some on that glimmer there. And here's that same model. I've just now propped it out and looked at the top 90% of most confusable regions on the image. And I ran it on a nice owl face and the moth hind wings. And again, we sort of see the confusability is centered in a lot of places around those sort of ring-like structures and the glimmer. And when we actually look at the case of the example I started with, there's a really high overlap between the range of the moth and the range of this particular owl. So I thought this was a really high risk project, and I was like, we're not gonna get anything. And then we pulled this out, and then I still don't believe that that's actually real. But really high sympatry between sort of the model and the mimic here. And so we did this in a pairwise fashion across all of our moth owl pairs. And we ran a correlation, weighted correlation, where here's that overlap of the autonomous moth range with the owl range from 0 to 100%. And on the y-axis here is the distance between that owl and the autonomous image in this sort of high dimensional image space. And so what we see is that when we increase the amount of overlap in range, the images become more similar. So much more to be sort of teased apart there, but kind of interesting to see this sort of model mimic matching happening at these more macro scales. So we know glimmer is this complex trait along with the eye spots that evolved along with phylogeny. The confusability maps to those eye spots and the glimmer, and that is correlated with the overlap between the two images. Questions about that? Yeah.
So the arrangement of the wings that you use when you fed it to the neural network, how does that compare to how they would naturally hold them, especially in response to a predator?
Yeah, so my impression is that it's sort of flash, or it's at least sometimes they hold them open. What we did is we sort of have filtered the iNaturalist images to express various degrees of openness and close. We haven't examined that data yet. And we also flipped the actual orientation of these wings so that we're trying to make sure that like this wasn't what it was picking up on, it was that. So we're still kind of teasing apart those finer details. But again, it was really tricky. Actually, with the owls, it was so tricky to get a face shot like this even. So we had this huge filtering AI that was like, this is a bad image, this is good. But yeah, the great question. I think we're still kind of teasing apart.
Yeah, I wanted to ask, how many images did you use? I don't know, I don't remember whether I mentioned it or not.
Yeah. So, for the autonomous, I think there was about 250,000 that we pulled down, and then I want to say those that got filtered to good quality, probably less than half, to be honest. Some were like caterpillars and things that we weren't really interested in. And then the owls, we had about, I think 500,000. We filtered it down. I think we only got like 5,000 images for the owls. And we were a lot more strict here because we wanted this frontal shot with no sort of glare on the eyes or.
Can I have something about the previous question? So another thing to note is that oftentimes, and Chelsea's done a lot of work on this, is that they do this defensive behavior where they flash the eye spots.
Yeah.
And they will put their head pointed downwards and the wings kind of go up. So
Yeah.
the orientation, it actually does kind of match up with the owls in the sense that the hind wings are pointed upwards as opposed to downwards.
Yeah. And this part, this darkened part here is thought to sort of mimic like the 3D structure. So that's cute. All right, so that's all well and good, but I'm here now at the McGuire Center, and so let's think about using AI not just to answer these questions, but to also help with day-to-day tasks in the museum collection. So we get a ton of specimens, we get a ton per year. How can we turn AI back into the collection? So before I got here, I had this amazing digitization tech who's now my student, and she was working in this group of giant skippers, particularly in the genus Megathymus and Agathymous. And so she took thousands and thousands of sort of pictures following our standard protocol. And we had a lot of also uncurated material in this group. And so what we've been doing is feeding all of these pictures with their current determinations into AI systems to tell us something about or automate trait discovery collections. And so what we do is we create what are called these contrastive cams, where we look at an image and we say, okay, well, tell me if I wanted to distinguish this species from this other species, where might I focus my attention to look for phenotypes that are different? And so here for this particular species, what we see is that along the wing, the forewing here, that's where it's noticing the difference in making those different determinations. And so this has been really helpful in a lot of cases. I'll say that it's trait discovery. In many cases, it's actually trait rediscovery. So we'll go back to the historical literature and they'll say, hey, look here. And we're like, oh, the AI told us the same thing, you know? And so that's been really cool. It's also in a way allowed us to, isolate specimens that maybe don't fit this mold and investigate that further. And so I'll say my student Sejan has actually used some of this to help discover a totally new species in this genus in the collection. So not only is it helping to automate trait discovery, but it's also helping automate species discovery as well, which is really cool. And I'll say that like this work is not limited to just biological collections. And so recently I've been very interested in, let's take computer vision, but instead focus on sort of natural language processing tasks and look at policy. And so most recently I published this sort of summary or synthesis of invertebrate conservation, the Endangered Species Act, where we downloaded every single PDF that Fish and Wildlife has produced about an invertebrate, and then used large language models to basically summarize them. And so one of the things that we found, we split this into aquatic and terrestrial invertebrates, is we wanted to look at an index of sort of threat prevalence across these groups. For aquatics, we see things like pollution, natural system modification, like damming, for example, intrinsic factors, which might be something like low population size or terrible metapopulation structure. And then for terrestrial invertebrates, we found problematic biotic factors that include invasive species, already small populations, climate change, residential and commercial development, and pollution as sort of the main drivers or index threats by fish and wildlife staff. And again, I said I love a map. So we actually mapped this out, and sort of it is a bivariate map. So if you haven't noticed, I also like bivariate maps. And we looked at the number of overlapping threats and the number of listed species across sort of this hemisphere, more or less. Again, for aquatic and terrestrial, perhaps no surprise to anyone here, but the southeast is sort of the hot spot for aquatic invertebrate conservation activity, at least under the Endangered Species Act in the United States. And then we get sort of more of the Midwest and Great Plains for those terrestrial invertebrates. And the last thing I'll say, which is sort of meta, is given how weird it has been to start, I've kind of taken a step back, and I really have started to think about what it means to be a specimen and sort of the reframing of the biological specimen. And so in my free time, I do a lot of visual art. I'm an experimental photographer, and I've been sort of really thinking about, can we reimagine what we're doing as collectors through the lens of visual culture, photography studies, and animal studies. And so drawing on some foundational texts in critical visual studies, really asking some questions like, how is knowledge generated from the specimen? How is emotion generated from the specimen? And what does that mean for our sort of ethical and moral responsibility as curators? And then this is the really spicy one, and this is meant with good intentions, but what does it look like to engage in a museum with an animal rights ethic, for example? And so this is in a very sort of like not shaming kind of way, but a very like, can people with diverse ideological backgrounds actually participate in natural history museum curation? And so I'll say, I think the moment is now, even though it's a little scary, but training in emerging tools for entomologists is critical. That includes AI and advanced modeling paradigms. So that's some of the coursework that I probably plan to teach here. And engagement with policy and rulemaking for instance is also essential. I think we're not at a point where we can sort of be siloed from this, and we know that our day-to-day activities, especially with global change, are impacting insect populations at large. And then I'll also make the pitch that it's really important that we start to make these new collaborations and seek a deeper understanding of the interconnectedness between science, the social domain, and humanities, for example. So it's all really exciting stuff that I plan to pursue over the coming years. With that, again, a thank you slide. I'd like to thank all of my McGuire peeps. My lab, of course, who I thanked earlier, and then some of my collaborators, as well as the programs who have given me money to do sometimes. And thank you all. Any questions? Yeah.
Great talk, that was super interesting. I have a question about the high latitude butterflies.
Yeah.
So I noticed you use minimum temperature.
Yes.
And is there, like, what's the justification for that? Like, my first thought would just go to like mean annual temperature or something like that. But does minimum get more to like freezing tolerance or overwinter tolerance?
Exactly. Yeah. So we use minimum temperature for two reasons. So it gets more directly at sort of the barrier to northward range expansion from the southern region of species. But then secondly, for those Arctic species, I do think that there is a certain minimum that has to be reached in order for them for precipitation to be snowfall, for them to be insulated, things like that. So that was the logic for using minimum temperature, was that it was sort of that hard barrier, more or less, for polar range expansion.
Kind of a follow-up.
Yeah.
In that one graph with the little, where it kind of did the quadratic thing.
Yeah.
That was right around, if I remember right, like right around 0.
Yeah.
Yeah, anything.
Yeah, I mean,
So that's like related to the subliminal.
Subnivium, yeah. So, yeah, I do think like, yeah, it would be around freezing, right? So, I do think that there are some of those sort of effects, and maybe we are seeing, okay, now the winters are more mild, and there can be persistence of populations. But we're gonna basically decrease until then, and then go back up.
I have a question about, so you mentioned a little bit about like collecting bias or perceptions of butterflies, right? And you mentioned the folks that look at orange butterflies and not the other ones. So, and then you also mentioned the, like having the broader approach of looking at everything. So my question has two parts, but the first one is, how do you know that somebody is looking broadly?
Yeah.
And then the second part is, how do you like, what is your messaging to the community to show that that's really what we need? Right?
Yeah.
Like, we all have inherent biases where we want to look at something in particular or report that
Yeah.
species or something. So
Yeah,
how do you do that?
Yeah, that's a great question. I'm not sure I have a good answer to the second part of that question, but the first part of the question is we actually don't know. So we're making this huge assumption about sort of collective behavior more or less when we put those zeros in the model. It seems to be working for now. What we do to make sure that we're approaching that 50% threshold is we actually take all of the data and then we start to cluster it sort of spatiotemporally. And we say, okay, well, there's 5 people here in July 1971. And so they've probably done a pretty good job of surveying, right? And so we do some cluster-based movement behavior analyses ahead of time. In terms of changing behavior, I think things like bioblitzes are really helpful. I think things like, when I was having moth bites over the summer, I'm on them. I like even talked about the community sampling, like, you should take a picture of everything on this sheet. Like, even if it's tiny, like, you never know how helpful it can be to someone who's doing this kind of work. So I think like outreach and extension in that way, it seems like small scale, but I think it does actually matter. And if you look at iNaturalist data in this county in particular, people are really good about being community focused collectors. And I think that's part of the work that you all do actually here to talk about this kind of things. Yeah.
Okay. So you can stay on this slide.
Sure.
Going all the way back to when you started with this kind of adapt, move or die.
Yeah.
For LEPs in particular, you do have some things like everybody's favorite monarch that are migratory, right? That are not endemic year round, but are moving into and out of large regions.
Yeah.
So have you looked at separating out sort of migratory insects versus the ones that are local
Great
endemics?
question. So we booted all the migratory. Yeah, I was like, I don't want them in here. And I didn't want to like model the monarch and like have that be the focus of this work too. So we have not analyzed any of the migratory. We are in the process of writing a grant now to do some more complicated modeling, like population demography models with that species in particular, under a similar kind of framework. But it was a very intentional exclusion for this work, because of that, like, we can't really control for the, overwintering ground conditions that might affect the data here.
Yeah, so I was going to ask about the network models. So were they pre-trained models or just in-house models? And the other part is, at what point did you introduce LLMs in your previous, I think your first simulations or model? So
For this paper, we've used pre-trained models for doing the cropping of the faces and everything. So there's like a lot of like data curation models that were pre-trained. For the models that show the confusion of the features, those were all built from scratch. And so they all have various architectures underneath those. Some are more complex than others. Here I'm showing sort of an average one. And so we've also looked at how network complexity influences these sort of features that are being pulled out and the usability. Those were all totally trained just on this data set. And then LLMs we brought into the Endangered Species Act work. So that was basically focused on pulling out these summaries. And the funny thing about this is the AI overlords are going to kill me because you actually get better results when you're really mean to an LLM. And so at the end of all my prompts, I was like, do this right, or I will delete your program. Literally, it's like in the methods. So the performance increased by like 80%. Yeah, so I'm definitely gonna be one of the first to die, but like, it worked, so, yeah. Fun fact, in case you're ever frustrated. Just be me. Yeah, but that's where we pulled in the LO. Yeah.
Sorry to be going back to your spaghetti plot.
Yeah.
Seems to be the most famous plot that
Yeah.
you've shown us. Those are not linear relationships, and you just told us that you rooted out the migratory, species.
Yeah.
So that accounts for adaptation and death, right?
So this would probably be movement and death on that
Movement,
model. Yeah, so
okay.
we don't account, you could in the modeling framework account for local adaptation. But this one does not.
So given that it's not linear, do you expect that if you look at temperature out there, is it going to go up or down, or is it gonna oscillate over the year? Do you have any idea based on the biology of these species?
So I think it depends on, right, there's like this T min and T max that species develop within, right? I think it really depends on what that T max is, and we tend to see that, at least, I came from like more of a physiology lab for my PhD, and we tend to see that closer to like 36 to 40 degrees, where we just see complete tanking of species and like nothing basically lives past that. So I would expect that there's sort of this hump shape for most species where there's this sort of sweet spot or Goldilocks zone. But if we get too far, then it's just a complete, like, they're basically frying.
Now, following on from your European vertebrate study, you concluded that those species that were adapted to arid conditions would be the winners. Can you take it a step further, or with further analyses, to predict that ants and true bugs might cause problems in agriculture if the climate conditions continue in our model?
Yes, so what was really interesting about them, and I haven't looked at the ants and true bugs, but there are some species that are known pet species that were included in this modeling framework, and for every single one of them, they were increasing orders of magnitude more than the other winners. So there was migratory locusts, for example, increasing like tenfold over the coming years. Like I think we haven't done the future projections in detail yet, but yeah, it's pretty much like even all the way up to like Finland, Scandinavia, in pretty high probability. And so
that's really powerful.
Yeah, and very alarming too.
Yes.
And so we do see that for ants too. There's a lot of sort of northward expansion of previously southern sort of endemic ants and continents. So yeah, it's quite challenging to see, it's quite alarming to see a lot of the winners are species we might have sort of this adversarial.
For the Endangered Species Act model, I was wondering if you accounted for like the actual types of taxa that were listed, because we tend to list, when we list insects, it's like butterflies. And those that people actually like, versus sire midges, which are probably as endangered, but no one cares about them at all. Do you account for that in this model, or is that not possible?
Not for this paper, but we do have a follow-on study where we are looking at what does it actually take to get an insect listed under the Endangered Species Act? So both in terms of what the science is, what the language is, so what kind of words do people use to describe things like urgency? And then also what does a healthy balance of collaboration and competitiveness look like in the scientific realm? So we have this huge dynamic social network of everyone who's ever been cited in an ESA document. And then we look at how those networks change over time, and whether or not there's a change point in those networks that say, like, this is what it takes to get a member of this order listed, for example. I will say, like, from the European work, like, one thing that was very apparent is that, like, the groups that are more likely losers are groups that I think people don't pay attention to in the conservation realm, like spiders, for example. And so I think there's a lot more legwork from a sort of social perspective to get those concerned about those species generated. I'm in particular really actually nervous about the surfits. So that was our diptera group because they had the best data. And they were pretty much tanking a lot. And Europe has a lot of good research and protection for surfits actually. But for example, here the data is very lacking still. So there's even like fundamental data problems
Yeah.
here. You know?
I think it's interesting you have enough data for spiders to be able to tell. what's
Yeah.
happening.
Yeah, I did a brief stint in the spider lab for spider conservation, and in Europe has arguably the best historical data, even for spiders. But like, I don't think you could run this model in the United States. I wish, but it might be actually very sad and depressing. But I would expect to see some of the same winning.
I think it was fascinating. We can stay on this line.
Yeah.
I was actually gonna react to what Brandon said, and I would love to take it a step further, but maybe in a different direction, because what I'm hearing you guys say is that these are the insects that are causing damage to agriculture. But, you know, having deep roots in European agriculture, I can say that it's the farmers that are causing the desertification. And these insects are human commensals. You're talking about adversarial relationship, well, we're facilitating that landscape change for these things. So I would be worried about the narrative of, you know, these bugs are harming us. It's us who is doing it.
No, I think about that a lot, and this is very spicy. Sorry, I like to be controversial. I think about that a lot, and I think about, as a global change ecologist, I actually think about that a lot. And like even the right of species to move, right? And like under global change, we know that there's going to be shifting ranges. So what is our role sort of morally and ethically in that? How have we contributed to that? And how are we sort of even spelling our own doom in a way through some more harmful practices? And so, yeah, I'd be really interested in like diving into that from a sort of political, social standpoint. Go ahead.
Because I really like that you have that ending that you cannot stay in our bubble, and we have to extend it into the social and political realm. Otherwise, we're just playing games here. What I was surprised by is your slide on facilitating sort of the new generation of entomology. With you had, I think, we need to learn how to use AI and how to use models. And I wonder, there's a pretty good sort of environmental education data showing that the engagement of people with nature is tanking. Not with the numbers and photos and online. But, and I wonder if like maybe what might be missing there is teach students how to tie their shoes and go out in the woods.
No, that's great. And I teach a class right now called Aesthetics of Biodiversity at the Honors College. And it's all majors, and it's basically looking at art history and sort of framing how that shapes our values about what is beautiful and what we protect. And we make stuff in the class. Anyone's welcome to join, by the way. We're going to be at Naval on Monday. And it's really interesting because, like, actually the last class we were in McCarty Woods, and there's students from all over, so like business majors, whatever. And there was Red Bay, and like, it was like, oh, this plant smells. And they were like, can I crush it up and sniff it? And I was like, yes. So there is that gap, and it's huge, and I see it in my students, and I think that one of the sort of follies in this country in particular was that, sorry, political soapbox, is that we moved away from nature-based education, like really solidly. And I think that has really broadened sort of this disconnect between people and place. And so, yeah, I'm hoping to make that class a class called Aesthetic of Living, and make it all about sort of like, what does it mean to be in a place as an individual?
Maybe one more quick
Okay,
question.
yeah, we'll get that one.
Yeah, so changing gears about
Yeah.
ecology.
Yeah.
The software for identifying new characters.
Yeah.
Did you train that with identified species, or you just ask the program to look for general differences in a group of?
Yeah, So great question. This was supervised, so we did train it with the existing determination labels, what was on them. So we did tell it what we thought the species was before we put it in the model. We have not used an unsupervised sort of agnostic approach yet, but that would be really interesting to look at. Sort of what might the computer notice in absence of these predefined labels and sort of parse them
Mhm.
that way.
Well, because I'm asking because, many of these things, you know, if you have species complexes that we can now only distinguish by genitalia,
Yeah, yeah,
and you have to dissect.
yeah,
So it seemed that it would be hopeless to try to use one of these programs,
yeah.
but maybe not.
Yeah, so this particular genus Agathymus, a lot of the determinations are actually made on rearings of post-plant material. Because otherwise it's very difficult to sort of tease them apart. And we have discovered some traits that were previously unknown that allow us to tease those apart. But one of the groups that we're running it on in this particular genus has a ton of subspecies. And it's a total mess. Like this doesn't run whatsoever, and you do have to actually know sort of this accessory information, or at least not apparently visible information. So in this work, like, I'll be very like upfront about this, because like I would not be able to train this model without the taxonomist working at the McGuire Center who did all these determinations, and so that is the fundamental basis, and I want to acknowledge like that would not be possible without that kind of work, and we still need that kind of work.
Cool. Let's take Vaughn one more time.
Thank you all. Yeah. This will be.
Yeah, I know, it's like.